Excitatory postsynaptic potentials. The combination of chemical transmitters like glutamate and aspartate with the receptors make the postsynaptic membrane partially depolarized. That is, there is a drop of potential across the membrane from, for example, minus 80 to minus 65 millivolts. This difference of plus 15 millivolts is called as excitatory postsynaptic potential, that is EPSP. The initial depolarizing response produced by single stimulus to the sensory input begins at 0.5 milliseconds after the afferent impulse enters the spinal cord, and then it reaches at its peak at 1 to 1.5 milliseconds later, then declines exponentially over the next 4 milliseconds if not sufficient to cause generation of the spike. During this period, the excitability of the neuron to the other stimuli is increased, and consequently the potential is called as EPSP. The excitatory postsynaptic potential makes the postsynaptic membrane hyperpolarized, and if sufficiently strong, it causes action potential to develop in the postsynaptic membrane. Ionic basis of development of EPSP. When action potential arises in the presynaptic knob, the synaptic neurotransmitter is released and it combines with the receptor of the postsynaptic membrane, and this combination causes opening of sodium or calcium ion channels in the postsynaptic membrane. The entry of sodium or other cations inside the cell from the extracellular fluid leads to hyperpolarization and development of EPSP in the postsynaptic membrane. EPSP can also be produced by agents that close potassium channels. Excitatory postsynaptic potential due to activity in one synaptic knob is small, but EPSPs produced by each of the active knob can summate, and this summation may be spatial or temporal summation. Spatial summation. On stimulation of two afferent fibers simultaneously, both which make synaptic connections with the motor neuron, their EPSPs get summated to the critical level, and thus they may produce spike discharge from the motor neuron, and this is called as spatial summation. In other words, the activity in one synaptic knob facilitates the activity in another to approach firing level. Temporal summation. If we stimulate an afferent nerve for the second time before the decay of first stimulation, neither of which alone can generate a spike from the motor neuron. However, each produces some EPSP which gets summated to the critical level necessary for the initiation of spike potential, and this is called as temporal summation. This is diagram showing the temporal summation and the spatial summation and the temporal summation. So here at the postsynaptic membrane, two different stimuli they produce different EPSPs, and they are going to summate to produce action potential, whereas temporal summation at one postsynaptic membrane, the at the single spot there will be multiple stimuli one after another. They are going to produce EPSPs, and these EPSPs they are going to summate, and if the potential it is going to reach to the threshold stimulus, then it is going to produce action potential. Inhibitory postsynaptic potential. Sometimes the combination of chemical transmitter like GABA or glycine in the brainstem and spinal cord with the receptors make the postsynaptic membrane hyperpolarized. For example, resting membrane potential is going to reach minus 90 from minus 80, and this difference of minus 10 millivolts is called as inhibitory postsynaptic potential or IPSP. Hyperpolarization begins at 1 to 1.5 milliseconds after the entry of afferent impulse into the spinal cord and rises to peak in 1 to 5 milliseconds after its onset, and declines exponentially with the time constant for about 3 milliseconds. During this potential, the excitability of the neuron to the other stimuli is decreased, and consequently it is called as inhibitory postsynaptic potential. Ionic basis of development of IPSP, inhibitory postsynaptic potential, is due to inhibitory chemical transmitter liberated on the subsynaptic membrane. Therefore, uh, the membrane becomes permeable for potassium and chloride, but retains high degree of impermeability to the sodium. This results in hyperpolarization of the whole postsynaptic membrane. IPSPs can also be produced by opening of potassium channels with the movement of potassium out of the postsynaptic cell. In addition, they can also be produced by closure of sodium and calcium channel. This is diagram showing of the IPSP, that is inhibitory postsynaptic potential, where sometimes membrane potential can reduce from -40 to -50.